everyone. My name is Anisabel Guzman, and I was in the Giovannoni lab this summer. And my project was identifying the genetic loci contributing to folate accumulation in tomato. So, folates, or collectively termed as tetrahydrofolates, are one of the most important vitamins for the human body. They are composed of a pterin, a p-aminobenzoic acid, and glutamate chains. For the folate biosynthesis pathway, it is split between three compartments, with the pterin branch found in the cytosol, the p-aminobenzoic acid found in the plastids, and the synthesis of folates occurring in the mitochondria. So folates are important because they are essential in one-carbon metabolism, and they serve as cofactors in the synthesis of DNA and certain amino acids such as methionine and serine. Plants and microorganisms can synthesize folates de novo. However, humans cannot produce folates on their own. Thus, the intake of foods like legumes, asparagus, leafy greens, and citrus fruits are important for bodily functions. Without these foods, it can result in a folate deficiency, and this can cause cardiovascular disease, certain cancers, anemia, and neurotube defects. Many countries have fortified their foods with synthetic folic acid, and to try to combat folate deficiency. However, too much intake of synthetic folic acid can result in too much in the blood plasma and can mask the symptoms of vitamin B12 deficiency. Folate biofortification is an alternative way to increase folate levels in fruits and other food crops naturally and help prevent folate deficiency and minimize the adverse effects of synthetic folic acid. Biofortification is the process by which the nutritional quality of food crops can be improved through breeding, biotechnology, and agronomic practices. This can be done to increase the folate content being accumulated in food crops and diversify the folate rich foods. On the blue table, it shows how there have been multiple experiments done to increase folates in other food crops. And as you can see for tomatoes, they were able to increase the folate content by 25-fold, but there is a buildup of the precursors. So why do we want to increase folates in tomatoes? Well, they are the second highest consumed fleshy fruit in the world. They are high in vitamin C, carotenoids, and flavonoids. However, they are low in folate content. And we study tomatoes because they are used as a gene function and understanding factors that affect maturity and ripening of fleshy fruits. This is due to their simple diploid genetics, their short growth cycle, the ease of transformation, and similarities between species. In order to utilize these characteristics of tomatoes, we use the Solanum pinelli introgression lines. These were derived from a cross between the green-fruited wild species Solanum pinelli and the Solanum lycopersicum cultivar. These crosses were done by Danny Zamir's lab. They consist of 76 lines and were generated through a series of self and successive backcrossing. Each line contained a genetically mapped single introgression from Solanum pinelli in the genetic background of the domesticated tomato M82. These lines have been previously used to identify favorable genes that can improve yield, disease resistance, and fruit quality. They can also determine the ability to modify fruit folate using the wild species and genetic diversity. So, before the summer, there was a pyramiding experiment used by my mentor, and pyramiding involves stacking one or multiple traits, in this case high folate content, into one genotype. And this was undertaken in the introgression lines 4-4 and 8-3. And it was done to ascertain if folate levels could be increased through pyramiding in the hybrid progeny of these lines, to study folate accumulation and determine the causal genes responsible for the increased folate content. We chose the 4-4 and 8-3 lines because the 4-4 contained a higher folate content and the 8-3 contained intermediate folate levels. The overall goal of the project is to develop tomatoes with high folate and identify what controls or regulates that pathway. My goal for the summer was to measure folate content found in the F3 population and genotype the F3 and F4 M82 cross populations. By doing this, it could possibly open up a way to use this knowledge onto other crops and increase dietary intake and range of dietary sources of folate. Our hypothesis is that pyramiding the introgression lines 4-4 and 8-3 will produce lines with elevated amounts of folate and other traits not seen in the parents. So before the summer, the initial experiment done by my mentor was to cross the 4-4 and 8-3 introgression lines. The resulting F2 lines were then grown in the greenhouse, and there were different phenotypes observed. They were taller, the fruit had green shoulders, unlike the parents. There was microfissuring. Um, the fruit tasted sweeter, the folate levels were higher than the control, and from this F2 population, we decided to choose number 16 and number 35 to continue on for the F3 population. So she started genotyping the data for the new F3 population, and genotyping comprises of extracting DNA from leaf samples of seedlings and screening with genetic markers. These markers flank the regions 4-4 and 8-3. Then the PCR was utilized to amplify specific regions using the CAPS markers. The amplicons are then digested by restriction enzymes, and the DNA fragments are separated by agarose gel electrophoresis. The gel was then visually analyzed and scored to determine the genotype for the corresponding genetic marker of each line. This helped determine the genotype of the hybrids at the corresponding locus in the chromosome and also access any recombination that may have occurred. This was the 16F3 population, and using the genetic markers bordering the introgression lines, we found a majority of hybrids that were heterozygous for the selected marker, but there were no introgression regions for the 8-3, and a few hybrids showed a change in a genotype. So this was the 35F3 population, and using the genetic markers, we found a majority of hybrids that were heterozygous as well, and a subset of the F3 lines had a double introgression. A few hybrids showed a change in genotype, indicating a recombination event. Then, when the summer started, I had the job of doing folate extraction, and I had the job of doing a folate extraction from the F3 population. And for folate extraction, the M82 cultivar, the 4-4 and 8-3 were used as controls. The fruit was harvested and frozen in liquid nitrogen. Then it was ground into powdered form. From then, these samples were extracted and measured using a microbiological assay. The assay uses growth of the folate-dependent bacteria, Lactobacillus rhamnosus, to assess the folate concentrations in a given sample. The assay's growth media lacks folate, so folate becomes a limiting factor of the growth of the bacteria, with the only possible source derived from the fruit extracts. On the left side of the screen, the initial folate results from the crosses in the greenhouse. These are the ones contained higher folate. A bigger F3 population was grown in the field, and the folate in the F3 population is shown on the right. On the right are the genotypes containing the Pinelli introgression lines, which are shown having a higher folate content. The homozygous lines also have a higher folate content than the M82. So on the left side again was the initial folate content for the 35F3 lines. It shows the folate content found in the greenhouse, and then the bigger F3 population on the right side. The ones containing the 4-4 introgression lines contained a higher folate content compared to the M82 control. So these are the candidate genes that are related to folate accumulation in tomatoes. 4-4 does not have any folate biosynthetic genes that we know of, but 8-3 does. It contains formal tetrahydrofolate deformylase, which is an enzyme of folate metabolism. It metabolizes 10 formal tetrahydrofolate to produce tetrahydrofolate. So for this portion of the project, we were able to find that the F3 population had a higher folate content compared to the F4-4 introgression region. This was from the pyramiding effect that we included both 8-3 and 4-4 into the crosses to increase folate content. We learned that the folate accumulation is a complex trait that is influenced by additional introgressed loci, or epistatic interactions. For the other portion of the project, based on the folate content in the select F3 lines, they were backcrossed to M82 to try to narrow down the region for folate accumulation. The F3 and F4 backcrossed 1 species lines from this population was grown in the field this summer. And from the initial crosses, the folate content was higher in the 35-4 lines. For the F3 and F4 BC1S population, they were germinated in the greenhouse and then transplanted to the Cornell Homer C. Thompson Vegetable Research Farm for the remainder of the summer. But before transplanting them in the field, I had to take initial meristematic samples of the plants to extract the DNA for genotyping. After figuring out which plants consist of genotypes of each parent, I then could map out which plants would go into the field. These are the lines that were genotyped and then transplanted to the field. A higher number of them are shown to be heterozygous for both parents, which can possibly result in a higher folate content, and also the ones with the Pinelli introgress region. These are some of the characteristics in the field currently. Um, a lot of the bushes are more compact rather than spreading out. The fruit has dark green shoulders, unlike the parents, and the fruit contains microfissuring, which are small little cracks in the fruit. Because this fruit is not yet ripe, for future work, we will need to check the folate levels of the fruit in the field. We will also need to check the expression of folate biosynthetic genes and potential candidate genes in select lines. By doing this research and figuring out the folate biosynthetic genes, we could possibly use this in commercial crops to increase folate content in other fruits. By increasing the folate content in commercial fruits, it can help combat folate deficiency and increase the diversity of folate-rich crops. I would like to thank Jim Giovannoni, my mentor Betsy, the whole Giovannoni lab, the BTI greenhouse staff, the Goderman greenhouse staff, the Thompson Research Farm staff, PGRP REU program, Delaney Sickler, Olivia Olston, and Natalie Henkos, BTI, Cornell University, NSF, and USDA. Thank you.